Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes, we have learned about how to do variant calling. We have discussed different variant types, and in the last class we have focused mostly on the SNVs and SNPs and small indels. Okay. So, we have talked about the pipeline that we use, different filtering approaches, etc. So, in this class the focus will be on doing the hands on analysis. Okay. So, we will utilize a tool to actually do this analysis ourselves, right, and we will do some variant calls. Okay. So, this is the topic that we will be covering in this class. So, let us go ahead. So, there are different variant calling tools. So, I will list some of them here. So, the first one is genome analysis toolkit. This is a very popular toolkit, and there are multiple options there. So, for example, you can have this GATK haplotype caller or GATK mutate2, right, and they are built for different purposes, although they have some overlaps, but they have some special purposes. We also have these SAM tools and BCF tools that are also widely used for variant calling, and then you have something called Freebase, Strelka, as well as FAIRSCAN2. Okay. So, out of these all tools, what we will use is the SAM tools and BCF tools. It is easy to use, it is free to install and easy to use through the command line. Okay. And since we have got the output in SAM file, we can use that for doing our analysis. Okay. So, what we will do is we will look into these SAM tools and BCF tool commands. Some of them, of course, there are many options, not all of them we require in this class, and also the time will not permit, and we will look at the command options that are available. Just to again remind you of the steps, right. So, we will follow these steps here, right, before we actually do the variant calling. So, we have the mapping data from the last hands on in the mapping. We will do the local realignment, we will do duplicate marking, we will not do base quality recalibration because of the time constraint, as well as it is a very computationally intensive process. So, we will not be able to do this in the lab in this short period of time, but you can of course try. I will mention the tools that we can use for doing this analysis, and once we have this analysis ready reads, we will use BCF tools to call the variants. Okay. So, let us get started because it will take a lot of time, because we will install these tools, SAM tools and BCF tools, and we will work with them. Okay. So, let us go ahead and see how to install them, and then we will come back to the steps that we need to follow for doing this variant calling. Okay. So, let us move to the terminal. Before I go, I will just wanted to show you these places where you can find these tools, and you can download. So, you have this genome analysis toolkit. So, this is the GATK, and you have this haplotype caller, etc. in here. Then if you go to SAM tools, right, or BCF tools, right, you will find them here. So, you can simply search SAM tools or follow the link that I have given, and you will end up in this page. Okay. And you can see these commands here for SAM tools, etc. And similarly for BCF tools, you will see manual, and this means you will have this command pages, right. So, there is a list of commands you can see here, and we will use some of these commands for our analysis. Okay. But before we go anywhere, we first need to install this, right. So, here if you just go to this link htslib.org, you will find this SAM tools, BCF tools and htslib. Okay. So, we just need to install these SAM tools and BCF tools, and we can download very easily here from GitHub, right, or here downloads you can go. So, we will get all these tools at once, and you can install them. Okay. So, I will just go through the installation process. I have downloaded them already, and we will go through the installation process, and then we will get to the variant calling part. Okay. So, let us go to the terminal here. We are in this place where we have this SAM files. Okay. We have generated the SAM files in the last lab, and you can see those SAM files here. So, let's see where the SAM file is, or we can simply search, right. It should be output SAM, right. Okay. So, this file is there. Okay. So, we will work in this directory, so that we can process the SAM file to identify the variants. Okay. So, I have downloaded this SAM tools here, you can see, right. So, this is SAM tools start.bz2, okay, and we have BCF tools again in the same format, and we have to extract this first. Again I have extracted. So, the extraction command you remember, right, tar minus zdxbf, right, and let us say SAM tools, right. So, once you run these commands for both the SAM tools and BCF tools, they will be extracted. Now, once they are extracted, you should see the folders, right. So, again you can see, right. So, you can see this folders. So, one is this BCF tools folder here, right, and you also have the SAM tools folder. So, inside this folder you have these programs, right, SAM tools and BCF tools, which you can use for running all sorts of commands. Okay. So, I will just run this and right. So, it will give you all the options how to run these commands and these things, right. So, usage. So, you can see the usage, right, as before. You have SAM tools, command and options. Okay. So, there are different commands that you can use here, they are under different operations, right. So, you can see indexing, or you can have editing, file operations, statistics, viewing, etc., right. So, we will use some of these commands, okay, and also with some options. So, the options are not given here because you will find them in the manual in the web page. Similarly, we can also run the BCF tools, and again it will show you what are the commands that you can run here, okay. And okay, again for different purposes you have these different commands, and this is the usage, right, BCF tools command argument, right. So, this kind of usage that we will have to go with, right. Okay. So, again there are a lot more options for each of these commands that are given in the web page. So, if you go to the manual page, right, in the website, we will see some of these options. Okay. So, I'll show you like some of these options for our analysis, but of course not everything. All right. So, let's once we have this, now one of the things you will probably notice is just calling them through this dot slash is actually quite cumbersome, right. Every time you have to direct them to the folder, right, to that folder where they have been extracted. So, if you go out of this directory and go somewhere else in this system, you have to point to that directory where SAM tools has been extracted and call the command SAM tools. Is there any way you can call SAM tools from anywhere in the system? So, it turns out there is, right. If you are familiar with Linux, you will know this, there is a way to do this by adding it to the path. Okay. So, if you want to add this to the path, you need to have administrative access. So, means sudo access, okay, and you have to modify this file where which we will call as bash profile. Okay. So, this is called bash profile, and it will ask for this password, because for administrative access you need to give the password, and what we would have to do is we would have to add this folder in the path here under this path variable. Okay. So, what I will do is I will actually add both of these SAM tools and BCF tools under this path in this file, and we can then call SAM tools and BCF tools anywhere. Okay. Okay. So, let us go inside this SAM tools folder and look at the path, right. So, what I will do is I will just copy this path, okay, and I will add this to the bash profile. The notation is exactly same, right. You can see this here, and simply I will just add this path. Okay. And similarly, I can do this for BCF tools as well, and I am using VI. So, this means it is very easy, right. We can simply copy and paste, and BCF tools is also installed under the same path, right. Just need to change the name here, BCF tools 1.17. Okay. So, the versions are same. All right, and everything remains the same. We save it, and we come out into the terminal. So, again, if you are not sure how to modify these files, please do not do that, okay, because this might end up causing problems for your system. Okay. All right. So, once you have done this, now one of the last commands that we want to run is source. So, it will refresh, it kind of refreshes, right, it reads the path, and once it does that, right, we can now come out of this folder and we can call SAM tools or BCF tools from anywhere. Okay. You see, I do not need to point to that directory where SAM tools has been extracted, right. So, we can simply call SAM tools from anywhere, right. If we, even if we are in the, let us say, desktop or NG's data analysis folder, we can just simply say SAM tools and the system will automatically point to that directory and call SAM tools. Okay. And similarly, we can also call BCF tools. All right. So, this kind of makes our life a bit easier, right. So, we have now got these tools installed and we can call them from anywhere in the system. Okay. So, we will use these SAM tools and BCF tools now to identify the variants in our data, the SAM file that we generated in the last hands on. Okay. So, let us now go to the presentation and we will see like the steps, multiple steps and the commands that we will have to use, right. So, again we can search for this command through the manual pages, etc. But it will of course will not be possible for us to complete the full variant calling pipeline in this class if we do that, right. So, what I have done is I have also compiled all those commands that will use for doing this variant call, and we will simply run them, and I will show you, right, that at the end we get the variants. Okay. So, there are multiple steps. So, if we have to do this by SAM tools and BCF tools, they relate to these earlier steps here, but they are specific for these programs. This kind of like what these programs will need. Okay. So, the first step is we need to convert this SAM file to a BAM file, okay, again in the binary format, because this is a machine readable format, right. So, SAM tools and BCF tools will take this format. We need to sort the BAM file. So, sorting means it will by default sort according to coordinates or mapping positions in the file. And the third step is the realignment, which as we have mentioned, SAM tools and BCF tools they cannot do realignment. So, we will rely on another tool. The fourth step is mark duplicates, right. So, we need to identify duplicates in the data. The fifth step is the base quality recalibration. Again, as I said, this will not do this step in this class because of the time constraints. It is a very computationally expensive process, and finally, we will do the variant calls. Okay. So, the idea is to give you like how you can proceed, and of course, you can try out different things. Okay. So, one of the things you should notice is that when we install SAM tools, etc., you might end up with some issues with some libraries. Okay. So, you would have to install them independently. Okay. For installing SAM tools and BCF tools, you have to again rely on their install commands that are there. So, I can go and show you these install files that actually mention step by step installation process. Okay. Okay. So, I will show that, and then we will start with the conversion of SAM to BAM. Okay. So, conversion of SAM to BAM it actually uses this kind of command. So, it uses this command SAM tools view minus SP. Again these options, if you go to the manual, you can see what these options are doing, and then this is the notation. You give the SAM file name, greater than sign, and the output BAM file name. Okay. What the output BAM file should be. Okay. This is the first step. This is conversion to SAM, conversion of SAM to BAM. Then you have sorting of BAM by coordinates. Okay. So, again this is the command SAM tools sort command. Okay. And then you have the option minus O. So, here the notation is slightly different. So, minus O means this is the output file name. So, sorted out dot BAM, this is the output file name, and the output dot BAM is the input file. Okay. So, let us now go into the terminal, otherwise you might kind of get confused with all these commands. Okay. So, we will start with this first one, which is the conversion of SAM to BAM. Okay. So, as I promised, right. So, inside these folders via SAM tools, you will have something called an install file, and this will describe everything that you require to install these programs. Okay. So, these are the system requirements for the libraries that are required. You might not have all these libraries installed, so you may have to install them. You will find like the SAM tools will give errors if you when you are trying to install them, if some of these are not present in your system. So, some of these you can very easily install using sudo apt install. We discussed this in the command line, right. So, if you, for example, if you want to install this, let us say zlib or libpz2, right, you can very simply do this using this sudo apt install libbz2, right. So that kind of option is there. So and the errors will tell you what you have to do, what kind of requirement is there, what kind of packages you need to install. Okay. So here are 3 commands that you need to execute. So the first one is /configure. So inside the SwayamTools folder, you have to go through this /configure, right. This will configure the program. Then you have make and then make install. So those who are familiar with Linux installation process, they will know like this is kind of a very common step for many programs. Okay. So this is what you have to do. Again, I have installed it already because otherwise it will take a bit of time to install these programs. So once you do that, you can then add it to the path, okay, that I just showed you. And similarly for BCF tools, you will also find this install folder, install file inside this folder, and it will again list out what you need, how you should process, what kind of system requirements are there, okay. And finally it will also show you the commands, right. So you have to move into the BCF tools directory after extraction. So cd, then run this /configure and make, okay. So these 2 commands are enough, you do not have to do again make install, okay. Once you have done this, you can add this to the path directory if you have administrative access, and you can call those commands from anywhere in the system. Okay, so I hope that clears up how to install these tools. There could be some errors, you probably will notice. I mean, this is something that might happen, and again there are workarounds those problems, right. So if it is simply just some library missing, etc., you can install those yourselves. Okay, so now we can move into the analysis part itself, right, after we have done the installation. Okay, so the analysis part is the first step, is the conversion, right, of SAM file to the BAM file. Okay, so the notation is this, right. So SwayamTools -sp output.sam greater than sign, and the output file name, this should be in BAM format, right. So you can see like what these options are is in the site. So if you go to, let's say, documentation, right, and how to's, right, this will, under this you will find all these tools that are here, manual pages, and you can go to the SwayamTools manual page and it will show you what these commands do. Okay. So right, so for example, yeah, so the first command that we are using is the view command, right. So the view command is here, okay, and if you click on this view command, you will see these options, okay, -b, -h, etc., right. So these kind of options you can go through and see what they actually do. Okay, of course we will not have time to discuss all these options, etc., but you can actually find them out, right, in detail. Okay, so the command is SwayamTools view, right, view -sp. So this will generate the BAM file. Okay, so we have generated the BAM file. You can check, right, whether we have the BAM file, and yes, the BAM file is there. We cannot see the BAM file. This is a binary format, right. We cannot see the BAM file or read the BAM file using VI, but we can use SwayamTools to actually read or at least see some of the data in the BAM file. Okay, so the command is again SwayamTools view output.bam, but we do not want to see the full, right. We just want to see the starting part, let's say, right. So we just type this head. So again I will repeat the command, SwayamTools view output.bam / head, right. So this head gives you the first few lines of this BAM file, right. Again, this is very similar to SAM file. Of course, we cannot read it using VI. Okay, so this is the first step done, right. We have generated the BAM file. Now we have to sort this. Okay, so if you go to the presentation, right, sorting this BAM file by coordinates. So there is a function called SwayamTools sort. We need to give the output name, right, and then the input file name. So the input file name is output.bam, and the new output file name is the sorted out.bam. Okay, so remember, right, after -o the output file name will come, and again you can see here, right, that there is this sort option. Okay, and how we call this is also given here, right. So how you should call this, and then again if you click on this you will find all the options that are available. Okay. So, let us now go there, SAM tools sort -O sorted out dot BAM output dot BAM. Okay. So, we have given the output file name after the -O, right, and this output dot BAM is the input file from the last step, right. So, we converted this and it will run, and then we will get this sorted out, right, and again we can see this by SAM tools view. And head, right. Okay. So, what you will notice now compared to the other ones, right, the earlier file is that this is now sorted according to the chromosome here, right. You can see this, right, most these reads they start with this chromosome number and the position wise they are also sorted, right. You have these 274, 275, 282, 310, they are sorted, right. So, this is sorted by coordinates. Okay, that is what this command will do by default. We will use another type of sort a bit later, and you will see we can also sort by read names. Okay, so that will be useful for the next steps. Okay, so once we have sorted, right, so we can go back to the presentation, right, and we have to do something called indexing. Okay, so why do you need this indexing? You will see in a moment. For realignment program, it will require this index file of the BAM files. Okay, so we will use this sorted out dot BAM for our realignment purpose. Okay, so realignment will require this index file for the BAM. In addition, if you want to visualize these BAM files, right, if you want to see like these alignments, this mapping against the reference genome, you also need the index file. So, coming to the next step, which is the realignment. Okay, so realignment cannot be done with SAM tools BCF tools, it does not have any such function, but we can use another standalone tool called ABRA2. Okay, so here is the link where you will find this package, and you can download and install, and you can use this. Okay, it's very simple. You just download one single file, the compiled version, you just simply run. Okay, it requires Java, and this is the command that we'll use. Again, I'll explain what this command actually means, and we'll run this command. So, again I have compiled this because it will be much faster to complete this process. Okay, so you can find out what each of these commands mean, and I can show you their GitHub page where these commands are also given, how you should run this command. Okay. So, let us now go to that, right. So, let us build the index and then do the realignment, and we will follow that with the next step. Okay. So, let us go to the terminal. Okay. So, for building index we simply use SAM tools index sorted out dot BAM. Okay. So, the index file you will see it will create, and you will see this, it ends with .bai. So, this is the index file of this BAM file. Okay. So, whenever you run this index command, it will create this .bam.bai. Okay. So, this file in the same directory, this is the index file. Okay. So, this index file is created now. We can then go for the realignment. Okay. And we will do the realignment using the ABRA2 package, and I will show you the package where it is. So, this is the GitHub link, right, where you find this package. Okay. And from here you can download this, right. So, on the right side you can see this releases, right. You can download the version 2.23, which I will use for this analysis. And down here, if you scroll down, you can see the commands that you have to use. Okay. And this is the this is the command, and it gives the input file, output file, the reference sequence, etc. And some of the temporary directories that you need to create before you can run this command. Okay. So, this is again explained in detail. You can read through, and you will understand how you should run this command. Okay. So, I will just simply run that command. Before that, we need to actually see whether we have this temporary directory created already. If not, we need to create that, and also we need to see this this file. This is the program that will execute whether this file is installed. Okay, so here you go. Yeah, so we have this version 2.23, right? So which we'll use for our analysis. Okay, so I'll simply, for the sake of time, right, I simply copy this command here, and I'll explain in a moment what this command actually means. Okay, so we are calling this program abra2. This is version 2.23, right? This XMX 6G means you are limiting memory usage to 6 GB. Again, you can reduce it. It will of course take more time depending on your system, right? If your system has only 4 GB, you might want to say like don't use more than 2 GB. Okay. So similarly, so in this system I'll use 6 GB. I'll say okay, we can use up to 6 GB. Then you have the minus in. This is the input file name, right, on which you want to run the realignment. Then this is the output file name, right. We have given an output file name sorted out.abra.bm. Then we have to give the reference genome sequence. Okay, so I'll give here the reference genome which I have copied here. This is the S28HC reference sequence which we used for mapping. Okay, then minus thread. So this is for speeding up the process depending on the system that you are working with, and minus tmp date. So this is a temporary directory that is required for storing some process, right, the program for this command. Okay, so I'm not sure if we have created this temp directory yet, right. So what I'll do, I'll check again, right. I'll cancel this command. I'll not run this. What I'll do, let's check whether we have created. No, so it's not there, right. So I'll create this and get the directory, and then now I'll run this command. Okay, and okay, so maybe it's okay. So maybe during the copying it didn't work out very well. So I'll run it again. Let's see, right, threads 4. Okay, so I'm seeing only part of it, right. So let's put the full command again, minus okay. So here we are, right. We are running this command minus xmx6g, limiting the memory usage, input file name, output file name, reference file name, threads, temporary directory, right, and the log file that will be created. Okay. Sorry, so the command is not correct again. So it's not, it's actually something to do with the copying part when you copy the command. So it should be Java, right. Okay, so let's, I think, start from here again, right. We think there's some issue. Okay, so we need to give the reference file name. So I've just copied the command. We need to specify the reference file name, which is stored here, S28HC reference.fsa. Okay, now it's running. You can see. All right. So here it will be done in a moment, and once this is done, we'll get the realignment done, and we can go for duplicate marking using SAM tools. Okay. So when it's running, let's talk about the duplicate marking part, right. It's running in the background. So in the duplicate marking, we have to first sort reads by name, right. So when you have to mark duplicates, what this program will do, it will say, okay, it will look for paired and reads, right, and their locations, and then compare against other paired and reads, right. So that's why you need to first do this sorting by read name. Then we have to use something called fix mate command, right. So, this will fill in mate coordinates and insert size, right. So, it will compare during duplicate marking, right. It will compare, the program will compare these mate coordinates and insert sizes, right, before it marks them. And after that is done, right. So, we have to again sort based on chromosome number and coordinates, and these will be used, right. So, for actual marking step. Okay. So, there are this actually is 4 steps right here. So, one is first we have to, after this realignment, we have to sort reads by name, right, using this command, and you see like we have used this sample sort, we simply need to use this -n option. Once we have done that, we have to use this fix mate command. So, it will identify the mates, it will fill in the mate coordinates and then calculate the insert sizes, etc. And once this is done, we have to again sort based on chromosome number and coordinates, because these duplicates will be identified by this position, by their positions in the reference sequence, right. So, as we have discussed, they should start at the same position and should end at the same position. And finally, the actual marking, right. This is the marked command that we can execute. Okay, so let us see if the realignment is done, and then we can do this step by step. Okay. So, this realignment is done, as you can see, and let us find out if this file has been created. Yes, this has been created. Okay. So, we cannot right now see what kind of changes have been made, right, because we have to visualize those changes. Now, what we will do is now we will run this actually, right. So, we will sort by the read names, okay, with the options option -n, right, that means the read name, sort by read name. Again, if you go to the options of SAM tools, you will find this, okay, and it should be done in a moment. Okay. So, the new file is now sorted to out.abra.bm. Okay. So, this BAM file, now we can go through the next step, which is the fix mate, right. So, this is the command, right. So, here is SAM tools fix mate, the option is -m. So, it will actually fill in the mate coordinates. So, that is why the -m option is required. Again, you will find this in the SAM tools manual. Of course, if you have to go through all this, it will take much longer if we have to find out what these options are doing. So, I am simply copying these options that we have that I have found out earlier. Okay. So, this is done. So, we can see that fixmate.bam has been created, okay. And once this fixmate is done, we can now sort based on the chromosome number and coordinates. So, that is the default sort here, right. So, this is the SAM tool sort -o sorted.bam, that is the output file name that we want to create now from the fixmate.bam, that is the input file name, okay, and it will run in a moment, right. So, see if this is done. Okay. So, here it is, right, sorted.bam is there, and we can now finally do the actual duplicate marking. Okay. So, let us do this, right. So, let us do the duplicate marking here, and this is the command mark dup, right, and -r means remove those duplicates, and -s means create small statistics, and the sorted.bam, that is the input file name, and then you have the final file.bam, that is the output file name. Okay. And this is very quick, as you can see, like there are only few duplicate pairs that have been identified, right. Duplicate total is about 38. So, that is okay, it is like out of the many reads we have like 50,000. So, 25,000 paired in, right. So, we have to throw out about 38. So, that is not a big number for this data, but if you are going with bigger data sets, these numbers will go up much higher. The next step is BQSR. Of course, as I mentioned, we will not discuss this, and I will just point you to this program based recalibrator that actually does this process for you. You can install this tool and try this yourself. And once we are done, the final step is the variant calls, and this step will do with BCF tools. Okay. So, there are two parts here, as you can see. The first part in this command is BCF tools mpileup. This is what you will have to do to the final file that we generated, the final PAM file. And again, you have these options that actually mention what is happening here, and then you have the second part which will actually do the variant calls. Okay. So, in this part you will see this -mv and then something called -ploid1. Okay. So, we are working with haploid genome here, not diploid genome. So, we have to mention this, otherwise it will consider this as a diploid genome, and it will generate this output in BCF format, and the output file name will be calls.bcf. So, -o is the output file name. So, calls.bcf. Again, you can find these options under the BCF tools manual page, right, what these commands are doing, how you should use them, what options you should use, and what these options mean. Okay. So, I am simply taking these commands and I will run them here. Okay. So, we need to define the reference file again, right. So, the reference file name would be, see, this is the reference genome that we used for mapping, right, and it should be done very soon. Okay. Now, one of the things it will tell you, right, this number of reads per input file set to -d250 means it is taking maximum depth as 250 or maximum coverage for each position as 250. Okay. So, that is fine. Then you have, once you have generated this calls.bcf, you can actually filter out or visualize these BCF files saying BCF tools view, and you can filter saying like I want only those variants that for which you have quality score greater than equals to 20. Okay. So, here it is, and once we type this, you see some of this list. Okay. So, this is really cumbersome, right. So, what we want to do, we want to write this in a file. Okay. And this is how we do this, right. We want to write this in this format called VCF .vcf. Okay. And we can then read that file and we can see what these mutations are, what these variants are, and how what are these type of variants are, right. So, whether they are SNPs or indels, etc. Okay. So, again the command is BCF tools convert, and we are filtering by this quality score. The output file name is filtered calls.vcf, right. This is not BCF, this is VCF, and then the original input file name, right, from which we are creating this output file. Why are we writing this VCF file? Because this is a human readable format. So, we can read it ourselves, right. We just simply open it with VI, right, and we can read all the data that is there, and you can again use this :AC NORAP. You can see these variants that have been identified from this data set. Okay. So, we will talk about this VCF format in the next class, this BCF and VCF, what these are and how this data is stored. So, we will discuss that in the next class. So, once you have done this up to this step, right, the final one step that is remaining is the visualization step, right. So, we want to do something called a manual check, where we want to identify these variants in the data and see whether these variants are actually there, right, and we will use this tool called integrated genome browser. So, that will be for the next class. So, we have learned how to do the variant calling ourselves, and we have seen this is a really multi-step analysis, and of course, within a very short period of time this is not possible to show all the steps, all the options that are available, and we can also customize these options based on data sets and requirements. Thank you.